Hello, welcome to Wednesday, September 23rd, 2020. I'm here with our science lesson for today. Make sure that you're answering the bell ringer on there. Should be 3 questions or so on there. Answer them up. Today we're doing comparing cells day A, which I know it's kind of weird because we did C and D first, but I wanted to start with animal cells. So today we are doing day A, which is focusing on the plant cell part. So bring up that slideshow that we've been working on. And we're going to be looking at some plant cells today. So the directions for each one of these are our microscope viewing slides in here this time around. You're going to draw and label certain parts of the cell. And at the end of the day, there's going to be some questions summarizing our plant cells that we saw here. The first one that we're going to see is a water leaf that is called Elodia. So it's going to be actually green underneath our microscope, so we're going to be able to see a couple of different things than the other day. First off, we got the cell wall instead of the cell membrane because it's a plant, so it's got a wall that's a little bit thicker. That's the thing that we're going to see instead of a cell membrane. Remember, it does have both, though. We're going to see the nucleus again. We're going to see cytoplasm again, but in this case, since we're dealing with a leaf, and there's going to be tons of these around, we're actually going to be able to see chloroplasts as well, and I'll show you what those are going to look like underneath our microscopes, but you have a microscopic image up above, too, of what that leaf is going to look like. So let me switch over to our microscope setup. So there's our microscope. I'm going to get it kind of zoomed in on here for things. There we go. And this is underneath low power. You can't really see anything underneath low power. For all of our drawings, we're going to draw it on high if we can. Medium is also acceptable. But here's going to be medium power. You just have to focus it in here. See if I can get it focused in. There we go. And here you can kind of already start to see the nuclei forming. We'll get a little bit closer with it, a little bit more precise. But we're able to see the nucleus. You can see some of the chloroplasts. I'll kind of point them out as we get a little bit deeper in the layer. All right, here we go. This is really in there. It's a little bit trickier to see just because of how things are looking, but we can see everything that we need to see. I'm going to use this middle one right in the middle and try to... this kind of one that's in the middle of your screen there. Let's see if I can open it up to let more light in. Nope, it is all the way opened as far as how much light is going to get into it. All right, but I'm going to use that one in the middle of the screen. Right in the middle there, you can kind of see the box appearance of them. The box is the cell wall that's surrounding them. Right around in the middle, there's that blob. We're actually able to see a little bit more. That blob itself in the middle is the nucleus with the nuclear membrane all the way around, and that actual dot inside of that is actually the nucleolus. So this is actually a clear enough sample there where you can see a lot more of the parts going on with it, but we just need to concern ourselves with the nucleus as a whole. Now, if you look very closely at the bottom of that middle cell, and you can also see it on the side there, there's these little tiny dots, kind of almost making a little arc right on one side. That is a line of chloroplasts just waiting for the leaf to use them. You can see it right there in the middle one. Also the one to the left of the middle also has some chloroplasts too there. You might have to hunt around for them a little bit. Let's see if I move this, if we can hunt and find. Usually towards the tip you'll see a little bit more, but it also gets a little bit fuzzier on the tip because those ones are a little bit more, roughed up when they're putting things in there. So you can kind of see some of the little dots on there. The little dots on these ones are going to be the chloroplasts in this one. So that's going to be our Elodia leaf and everything that you would need to label there. And transition back over to our slides. Our second one is going to be an onion cell. And we're making the onion cells fresh, and because this is Monday that I'm recording them, I do not have a fresh slide for you to view at this time, so I'm just going to go off the picture that's on the slide here. We're not going to be able to see everything on the onion that we did with the leaf, mainly because the onion grows underground and therefore does not have any chloroplast as far as the part that we see goes. So when you look at the ones today, like is up in the picture in the corner, we're going to be able to see the nucleus still, and you can see that dot kind of in there. You can see the cell wall, kind of the brick shape. They're more stretched out rectangles just because that's how the onion cell kind of develops. It's a little bit different. And then we would have the cytoplasm is all that clear area in there. The ones that we're going to get is made with this brown chemical called iodine. It helps stain, there it is. It helps stain the actual cell itself so we can see it. So I'll be making those fresh that morning so we have a fresh cell to look at. It's still going to be dead because an onion is still dead because it's removed from the ground. So it's not living anymore, but we'll be able to see kind of fresh cells, what they freshly look like. Same underneath the powers. You're just not able to do the chloroplast this time around when it comes to it. So you put your onion drawing with the labels on there. You can just use that slide and draw out a picture of that if you're not able to join us this week. And then there are 7 questions to answer at the end of plant cells. So answer them up. Don't have to use complete sentences about a complete thought. And that is the end of day A. Stay tuned for tomorrow. On Thursday, we'll walk through what part B looks like, or day B. With that, if there's any questions, do not hesitate to ever ask. Send me an email. Otherwise, hope everything's going fine for you out there, and we will see you tomorrow.